Some peptides and lipids generated in the endocytic pathway can be bound by MHC class I-like molecules that are encoded outside the MHC. Some MHC class I-like genes map outside the MHC region. One family, called CD1, expressed on dendritic cells and monocytes, as well as some thymocytes, functions in antigen presentation to T cells, but the molecules it encodes have two features that distinguish them from classical MHC class I molecules. The first is that the CD1 molecule, although similar to MHC class I molecules in its subunit organization and association with beta-2 microglobulin, behaves like an MHC class II molecule. It is not retained within the endoplasmic reticulum by association with the TAP complex, but is targeted to vesicles where it binds its peptide ligand. The peptide antigens bound by CD1 are therefore derived from the breakdown of extracellular proteins within acidified endosomal compartments and, like the peptides that bind to MHC class II molecules, tend to be longer than the peptides that bind to classical MHC class I molecules. The second unusual feature of CD1 molecules is that they are able to bind and present glycolipids, in particular the mycobacterial membrane components, mycolic acid, glucose monomycolate, phosphoinositol mannosides, and lipoarabinomannan. These are derived either from internalized mycobacteria or from the uptake of lipoarabinomanans by the mannose receptor that is expressed by many phagocytic cells. These ligands will thus be delivered into the endocytic pathway where they can be bound by CD1 molecules. The relationship between the peptide binding and lipid binding capacities of CD1 molecules is not clear. Structural studies show that the CD1 molecule has a deep binding groove into which the glycolipid antigens bind. Whether the peptide antigens also bind in this deep groove is not yet known. Although CD1 binding peptides are predominantly hydrophobic in character, it is thought unlikely that they bind to the same site as the lipids. It appears that the CD1 genes have evolved as a separate lineage of antigen-presenting molecules able to present microbial lipids and glycolipids as well as a subset of peptides to T cells. And that some peptides and lipids generated in the endocytic pathway can be bound by MHC class I-like molecules that are encoded outside the MHC. Have a great day.